Well, welcome to this talk on the liver, which is the largest solid organ in the body, with only the skin having a greater total mass. But in terms of the condensed tissue altogether in one place, the liver is the largest organ. And in men it normally weighs about 1.4 to 1.8 kilograms, and about 1.2 to about 1.4, slightly less in women. And the liver is involved in many of the biochemical processes which are necessary within the body. And this is why the liver is often referred to as the chemical factory of the body. So if ever you're talking about a biochemical process and you're not quite sure where that process is going on, it's most likely that it's occurring in the liver, this biochemical factory of the body. So anatomically, we can see the liver occupies most of the upper right quadrant of the abdominal cavity. Rounded upper surfaces fit snugly under the domed diaphragm, but the lower surfaces are more tapered, giving a fairly wedge-shaped appearance from the side. Now, young children have a much bigger liver relative to the size of their body. And this explains why the upper abdomen protrudes in young children, because relative to the size of the body, their liver is disproportionately large. And we see on this diagram on the left that the liver is in the upper right, and then on the upper left we have the stomach, and also tucked under there that we don't see in the upper left quadrant of the abdomen we have the spleen. Now in the diagram on your right, we've taken away the lungs, and we can see the blue line of the diaphragm separating the thoracic cavity above and the abdominal cavity below. And we can see the rounded top of the liver fitting neatly underneath the diaphragm. And we can also clearly see there from the cross-section of the ribs that the liver is somewhat protected by the ribs of the thoracic cage, particularly the right-sided ribs. We notice the two large lobes of the liver, the right lobe and the left lobe, and just tucked under there in green, I think you can probably see the tip of the gallbladder. Now, it is accurate to classify the liver as an exocrine gland. That's one of its functions, because it produces about 500 mils of bile per day. And this bile is secreted from the liver via the right and left hepatic ducts, and these merge to form the common hepatic duct. It's the cystic duct that transports the bile from the common hepatic duct to the gallbladder, and in the gallbladder the bile is stored and concentrated. And then the same cystic duct carries the bile out of the gallbladder into the common bile duct. Now, the pancreatic duct merges with the common bile duct before it communicates with the duodenum. And this arrangement means that the bile and the pancreatic juice both enter the duodenum together via the hepatopancreatic sphincter, what used to be called the sphincter of Oddi. Now, the liver is receiving about 70% of its blood supply from the hepatic portal vein, the vein which drains blood directly from the gastrointestinal tract to the liver. And on average, slightly over a litre of blood per minute passes into the liver from the hepatic portal vein. But in order to support metabolism, liver cells also need a supply of oxygen-rich blood. And this comes from the hepatic artery. So the other 30% of the blood supply to the liver is coming via the hepatic artery. Now, the blood entering the liver from the hepatic portal vein is only partly deoxygenated. It is still supplying some oxygen. But remember, the blood entering the liver via the hepatic portal vein has already circulated through the wall of the gut, so we'll have lower concentrations of oxygen. So to top that up, oxygen-rich blood from the hepatic artery also enters the liver to perfuse the liver with oxygen-rich blood for all these processes of metabolism that are going on in the liver. Because the liver is a very metabolically active organ, therefore needs a large oxygen supply. After circulating through the liver, venous blood is drained into the inferior vena cava via 3 hepatic veins. Now, the hepatic portal vein and the hepatic veins should not be confused. The hepatic portal vein carries blood to the liver from the gut, whereas the hepatic veins drain blood from the liver back into the systemic venous return of the inferior vena cava. So in this diagram we see that blood is leaving the heart via the aorta, and the hepatic artery is going to be a branch off the aorta. So the oxygen-rich arterial blood from the left side of the heart is entering the liver via the aorta and the hepatic artery. But there's also blood entering the liver via the gut. So you can see the gut at the bottom. The gut is drained by the hepatic portal vein, and that is taking nutrient-rich blood from the gut into the liver. And that blood going to the liver from the gut will also contain bacterial breakdown products going to the liver to be metabolized before they enter the systemic circulation. When the blood from the hepatic artery and the hepatic portal vein have been perfused through the liver, when they've passed through the liver, then all that blood will drain in the 3 hepatic veins, represented by 1 vein on this simplified diagram. And also on this diagram, I've drawn a dark arrow going down the way from the liver to the gut, which is the direction of bile. So the bile is produced in the liver, going down through the bile ducts and the gallbladder, also entering the gut. Now, the liver is actually composed of 4 lobes, but only 2, as we've seen, are visible from the front. The ones that are visible from the front are simply called the right and the left lobe, the right being significantly larger. But from the back we can see the caudate lobe and the quadrate lobe. And also, looking at the liver from the back, as we are doing in this photograph, we see that the left lobe is on the left and the right lobe is on the right as we view, because we're now looking from the back, of course. And the underside of the liver is beautifully sculpted to facilitate the way it sits on adjacent inferior organs. When we say inferior, of course, we mean below. So we notice the gastric impression on the underside of the left lobe, which of course is where the stomach will sit. And we see the renal impression on the right lobe for the top of the right kidney. Now we also notice the inferior vena cava on this model, and underneath this section of the inferior vena cava, if we could see through the inferior vena cava, we would see the 3 hepatic veins draining into that section of the inferior vena cava. And we also notice the hepatic portal vein carrying blood into the liver and the hepatic artery carrying blood into the liver. Remember, the hepatic portal vein is carrying in about 70% of the total blood supply to the liver. And looking at the underside, we also see the cystic duct, the common bile duct, the common hepatic duct, and the gallbladder itself tucked neatly underneath the liver. Now, we've noted that the liver has 4 lobes, but the liver also contains thousands of lobules. So within the lobes of the liver are numerous lobules, separated from each other by connective tissue. And these lobules are usually 5 or 6-sided structures, and they are cylindrical. At the corners of the lobule are branches of the hepatic portal vein, hepatic artery and bile ducts. And collectively these 3 vessels are referred to as a triad. And a normal liver contains between 50,000 and 100,000 lobule units. And an individual lobule unit varies in diameter from about 0.8 to 2 millimeters across. So these diagrams are starting to explain the histology of the liver, the way the liver is actually arranged at an increasingly smaller scale. So the top left we have the diagram of the liver, and the top right we have the diagram of the liver or hepatic lobules. And then the diagram on the bottom right is a magnification of an individual hepatic lobule. Now it's the hepatic lobules that are the functional units of the liver. The individual liver cells are called hepatocytes. And in an individual lobule, hepatocytes are arranged around a central vein, like spokes around a hub in a wheel. It's a radial arrangement of cells, as we can see in the bottom right diagram. Typically, the hepatocytes are arranged in a row, 2 cells thick. And these 2 rows of cells are described as a hepatic cellular plate. So we can see these hepatic cellular plates within an individual lobule in the bottom right-hand diagram. What we're looking at here is a simplified slice of a lobule. So if I'd drawn this diagram properly, these would radiate all the way around the central vein, but I've just drawn one slice. And blood enters from small branches of the hepatic portal vein and the hepatic artery. So that's the blood going in. And the arrows indicate the direction of blood flow from the outside of the lobule towards the central vein of the lobule. So the sinusoids are in essence the hepatic capillaries. In between 2 hepatic cellular plates are the blood sinusoids. And as we say, these are the liver's version of blood capillaries. So a sinusoid is essentially a type of blood capillary with very porous walls to allow more contact than usual between the blood and the tissue cells. And this increased permeability of the liver sinusoids is explained by the large pores found in the cells, which make up the endothelial vascular wall. And these large pores are referred to as fenestrations, explaining why liver sinusoids are sometimes referred to as fenestrated capillaries. Now, unlike other capillaries, sinusoids receive blood from 2 sources. Firstly, from a branch of the hepatic portal vein, bringing blood from the gastrointestinal tract or the gut. And secondly, from a branch of the hepatic artery. And this arrangement means venous blood and arterial blood mixes in the sinusoids as it passes through a lobule, from the outside of the lobule towards the central vein at the centre of the lobule. So hepatocytes are therefore supplied with nutrient-rich blood from the hepatic portal vein and oxygenated and oxygen-rich blood from the hepatic artery. Hepatocytes lining the sinusoids are able to selectively absorb substances from the blood, carry out chemical processes, and then secrete the products back into the blood of the same sinusoid. In the centre of each lobule are small tributaries of the hepatic vein. Remember, it's the hepatic vein taking blood from the liver back to the systemic circulation. Blood from the sinusoids drains directly into this central lobular vein. And this vein joins up with other lobular veins, which eventually become one of the 3 large hepatic veins, taking the blood back from the liver to the inferior vena cava and therefore back into the systemic circulation. So we've noted that in the liver sinusoids, the blood comes into very close contact with the hepatocytes, the liver cells. But as well as that, in the sinusoids, there's macrophages, because there's a hepatic macrophage system. Now, liver sinusoids contain these macrophages lining the walls. They have been called various things in the past. Kumpfner cells or part of the reticuloendothelial system are old-fashioned names that you might come across. But basically they're macrophages. These are the large phagocytic white cells that derive from monocytes. Now, like their counterpart macrophages in the spleen, the liver macrophages will also break down some old red blood cells, recycling amino acids and excreting the bilirubin. But most of this process takes place in the spleen, but some does take place in the liver sinusoids. But in addition, the macrophages in the liver perform a vital immunological function. Because if you think about it, quite a lot of the blood that's going into the liver sinusoids is coming from the hepatic portal vein. And the hepatic portal vein is draining the gastrointestinal tract, which of course is full of bacteria. So some bacteria will get into the hepatic portal vein and be transported to the liver. Now, it's vital these bacteria don't get into the systemic blood supply, because there they could cause bacteremia and potential sepsis. So as the blood from the hepatic portal vein goes through the sinusoids, any bacteria in that blood will be detected by these hepatic macrophages, and they will phagocytose the bacteria. So the liver sinusoids, if you like, are a first line of defence to stop bacteria from the gastrointestinal tract getting into the systemic circulation. They're strategically placed to take the bacteria from this blood that's coming from the hepatic portal vein, draining the gastrointestinal system before it gets into the systemic venous circulation. What we're looking at here is a cross-section of part of a liver lobule. One of these sort of slices, a fairly thin slice in this case, of liver lobule. And this shows two hepatic cellular plates, that's two lines of hepatocytes, with blood flowing through the sinusoid in between. So you can see the branch of the hepatic portal vein, the branch of the hepatic artery, the central vein in the middle of the lobule, and the blood flowing towards the central vein. That bit we know already. Now, individual hepatocytes excrete bile into the caniculi, which then flows between the two rows of cells to be collected in a small bile duct. So the caniculi, here we see one caniculus labelled, are the bile channels. So the bile is produced in the individual liver cells, in the individual hepatocytes, and excreted into these channels, these caniculi. So the bile, of course, is flowing in the opposite direction towards bile collection channels that ultimately will collect together into the right and the left hepatic duct to export the bile from the liver. Now, this diagram on the bottom left is a transverse section of the two rows of cells which form a hepatic cellular plate, the two rows of hepatocytes. And the caniculus is actually formed between the two rows of cells. So bile produced in the individual hepatocytes is only excreted into one caniculi. This means bile is only excreted from one side of the hepatocyte, as the other side of the hepatocyte is in contact with the blood. And arrows indicate the direction of bile flow. So the bile is being produced in the hepatocytes, or processed in the hepatocytes, entering the bile channels to flow towards the bile collection channel that we can see in the diagram on the right hand side of this slide. So between the two rows of hepatocytes are microscopic channels called caniculi. Individual hepatocytes produce bile which they excrete from the cells into these caniculi. These in turn drain into the small bile channels of the triads. And the triads are the group of the three vessels we can see on the outer part of the lobule. So a triad is going to consist of a branch of the hepatic artery, a branch of the hepatic portal vein, and the bile collecting channel. So these in turn drain into small bile channels of the triads, which, as we've said, are tributaries of the larger hepatic bile ducts. And this arrangement means that bile flow in caniculi is in the opposite direction to the blood, which is passing through the sinusoids. So the blood in the sinusoids is going from the outside of the lobule in. The bile which is produced by the hepatocytes is going in the direction from the inside of the lobule out. The separation of the sinusoids, that's the blood channels, and the caniculi, that's the bile channels, prevent bile and blood mixing. And of course that's very important, because if bile gets into the blood, you probably know what that causes. Bile in the blood will lead to jaundice. But once formed in the physiological situation, bile flows into progressively larger bile ducts, which eventually unite to form the right and the left hepatic duct, and then the common hepatic duct, and then into the gallbladder, and the rest of the story you remember from a previous slide. In this diagram, which is a longitudinal section, it can be seen how a caniculus is formed between two rows of hepatocyte cells. The dark arrows indicate bile flow, the white arrows blood flow. So the blood is flowing from the triad towards the lobular central vein, and the bile is flowing away from the lobular central vein towards the triad. And as we can see, the bile is being collected in small bile ducts, and the blood is entering the sinusoid. And although the branch of the hepatic portal vein and hepatic artery are not illustrated in this diagram, we know that that is where the blood from the sinusoid comes from the hepatic artery and the hepatic portal vein. Now this diagram looks a little complicated on first inspection, but it's worth spending a bit of time to try and work out what it's saying, because it's an illustration of the flow of tissue fluid through the perisinusoidal space of Dis. Now the perisinusoidal space is what it says. It's the space around the sinusoid, around the liver capillary, the space of Dis. Now tissue fluid, which is high in protein, is formed because, remember, the capillaries in the liver, the sinusoids, have very large fenestrations in the endothelial vascular cells that make up the sinusoids. So there's very large gaps in the vascular endothelial cells which comprise the wall of the sinusoid, which comprise the wall of the liver capillary. So protein-rich fluid is able to get through. Now, normally in systemic capillaries, the gaps in the wall of the capillary are fairly small, and we don't expect many proteins to escape. But in the liver, it's large, the fenestrations are large, so the larger protein molecules can escape from the sinusoids. So this fluid goes into this perisinusoidal space of Dis, and this fluid circulates over the hepatocytes. This allows substances from the tissue fluid to enter the hepatocytes. Substances which have been formed by the hepatocytes are also able to pass into the tissue fluid. So this tissue fluid is a good opportunity for exchange of materials from the tissue fluid into the hepatocytes, from the hepatocytes into the tissue fluid. And after circulating over the hepatocytes, the fluid is drained into lymphatic drainage vessels as lymph, as we would expect. And in this diagram, white arrows indicate the direction of the blood flow, and the small dark arrows indicate the direction of tissue fluid flow, the tissue fluid which will become lymph once it enters the lymphatic vessels. Larger dark central arrows indicate the direction of bile flow through the caniculus into a bile collecting duct. So we see that as well as the flow of blood and the flow of bile, there's also this flow of tissue fluid through this perisinusoidal space. So the pores, that is the fenestrations in the hepatic sinusoids, that is the liver capillaries, are much larger than in normal blood capillaries. This allows more water from the plasma than normal to enter the tissue fluid, which bathes the individual hepatocytes. In addition, the large size of the fenestrations allows the passage of protein from the plasma into the tissue fluids. Because remember, proteins are large molecules, but they can now escape through the large fenestrations. And this is in contrast to capillaries in other parts of the body which are essentially impervious to plasma proteins. Now, in the liver, there is a larger space between the walls of the sinusoids and the tissue cells than is normal in most other tissues. So again, the liver is somewhat unusual in this respect. This space between the capillary wall and the hepatocytes is called the perisinusoidal space, or the perisinusoidal space of Dis. Once formed from the plasma, tissue fluid circulates through the perisinusoidal space, bathing the individual hepatocytes. Now there's actually microvilli from the hepatocytes that extend into the perisinusoidal space to increase the surface area within the circulating tissue fluid. And this allows for free exchange of substances between the tissue fluid and the hepatocytes. So the hepatocytes are taking full advantage of this situation to be able to absorb from the tissue fluid, to be able to excrete things into the tissue fluid. And they take full advantage by having microvilli which increase their surface area for absorption and secretion. Now, after circulating over the hepatocytes, the tissue fluid is drained as lymphatic fluid or lymph into lymphatic vessels. From here, the lymph drains into the main systemic lymphatic vessels before it's returned to the blood. And about half of the volume of lymphatic fluid formed in the body is actually derived from the liver. So we see the liver produces a lot of lymph. So here we see a normal human liver with the right lobe and the left lobe. And of course, this is exactly what we've been discussing in this talk, the normal structure and function of the liver. And I hope you've got some appreciation now of the fact that yes, it is the hepatocyte, the individual liver cell, that is the functional unit of the liver. That's where the biochemical processing is going on inside the hepatocyte. But isn't there just this amazing infrastructure to supply it with nutrients, to supply it with oxygen, to remove the bile, to remove the lymphatic fluid and everything that optimizes the conditions for the physiological activity of these metabolically very active individual liver hepatocytes. Now that's the end of this first talk on the structure and function of the liver, or the way the liver is working and trying to relate the histology and the structures to the function. In the next talk, we're going to be looking specifically at individual liver functions. What is it that the liver does? And we'll see that the liver has a very wide spectrum of biochemical and physiological activity.